Very warm welcome to everybody who is joining our session today. Our session is entitled Educating the Researcher, Traceable Sample Management and Processing. My name is Stacy Renke. I am the chair of the Education and Training Committee for the Metabolomics Society, and I'll be your moderator for today's podcast session. Before we begin, I'd like to give you a little bit of background about this podcast-style webinar series. Metabolomics is a multidisciplinary field of science, and as many of you know, by its nature, it can't be standardized. So this means that researchers need to use a high level of judgment and critical thinking when they're performing their studies. So recognizing this, the Education and Training Committee of the Metabolomics Society created this podcast-style webinar series where we ask an expert panel to discuss the challenges, strengths, and weaknesses of different approaches, their decision-making experiences, and best practices from their point of view. So today's podcast-style webinar, we will have 3 expert panelists from both academia and industry, and they're going to share their experiences and recommendations. So the topic today, as I mentioned, is sample management and processing. So essentially, when the samples arrive in the lab, what do you need to do to plan your acquisition? What do you need to do to process your samples and deal with your samples? And also, equally importantly, how do you keep track of everything? So I would like to pass it over to each of the panelists to introduce themselves now one by one. So I'll ask you to introduce yourself, your experience in metabolomics, and what area you work in, if it's academia or industry. So Maria, I'll start with you first.
Okay, thank you, Stacy. Thank you for the invitation of being here. It's really an honor. Well, my name is Maria Eugenia Monge. I'm a research scientist from CONICET, which is the National Research Center from Argentina, the National Research Council of Argentina. My experience in metabolomics started in 2012 when I was working at Georgia Tech at the research group of Facundo Fernandez in the States. And then I came back to the country in 2014. I was recruited by CONICET to start a new lab, in a new research group, in a new research center in the country, and in a new research field for my country. So there were several challenges to overcome, and my main expertise is in mass spectrometry-based and target metabolomics. We also do some target analysis, and we also provide services, not metabolomics, but services to the academia using the mass spectrometers that we have in the lab. And that's more or less my... Thank you.
Thank you, Maria. Oscar, can you introduce yourself, please?
Hello, everyone, and thank you for the invitation. My name is Oscar Llanes, and I've been doing metabolomics since 2006-07 at the Scripps Research Institute with Gary Siusdak, and then I came back to Spain in 2011 to start with a new research group at the Universitat Rovira i Virgili in Tarragona. And I'm also heading or coordinating a metabolomics platform. So I have this dual hat of research and metabolomics services, although the services are mainly academic collaborations. And we have this research and service. We do a lot of bioinformatic development and wet lab with the implementation of different strategies, analytical strategies and data analysis strategies for the metabolomics platform. And yeah, basically my main expertise is on untargeted metabolomics. I've been doing mass spectrometry for more than 20 years now. Yeah, I think that's a good summary.
Thank you, Oscar. And Annie.
Hi, Annie Evans. I'm the Senior Director of and Head of Core Research at Metabolon, where I have been for 19 years. So I got my PhD in 2004. I actually got my PhD doing biological mass spectrometry, but in proteomics. So got to enter the world of small molecule back in 2004 with Metabolon. It was very startup back in then. I was the 12th employee. So when I first started, I was running the LCs and doing the QCs and figuring out how to prep a sample and how to do the informatics approaches. So it's been a long and wonderful journey on the course of my metabolomics career. Similar to Maria and Oscar, most of my experience is on targeted metabolomics, but also run the targeted metabolomics and lipidomics labs in both areas. Again, mostly doing research, which means I get to come in every day and play because, just trying to make everything better and make the process run smoother and capture all those gaps in the process. So that's a little bit about me.
Great, thank you, Annie. And so thank you and welcome to our three panelists. And our audience will also notice that we have another member who's joining us. So I would like to thank EMN committee member Susanna Palma for helping us out with this webinar session. Now, we do aim for this session to be fairly interactive, so we are going to start to pose a few questions that the EMN has provided to us, but we do want the audience to feel free to ask questions as well. You will notice at the bottom of your screen, there's a little Q&A box, so please do feel free to pose your questions, and we will try our best to have the panelists answer them. All right, well, without further ado, we will get straight into our questions. So the first question is, why is it so important to develop a system for sample handling and processing? So Oscar, I might direct this question to you first.
Well, I can give you of course, my experience over all these years. I have to say that we do not deal with very large cohorts of samples. So typically I have to say that the largest untargeted study, it's been around 500 samples. We have had larger set of samples for the validation, like 2,000 samples. Once we discovered metabolites in the discovery phase with untargeted metabolomics, we had to validate it in a targeted way with more than 2,000 samples. But we typically work in the order of tens and a few hundreds of samples for untargeted studies. So that is something I think important to highlight, because that is conditioning a little bit how we work in the lab in our university. The first thing we do is always to have a meeting with our collaborators, particularly when they are medical doctors. That's probably the most critical, because my experience is that they typically have samples in the biobank that they have little control sometimes on them. They don't even know how many defrost cycles these samples have suffered. And that's one of the things that maybe we can discuss later. So there is a lot of working before we receive the samples, a lot of checking, a lot of experimental design, and there is a lot of things that we have to double check before accepting samples and before we spend time and resources on these samples. Now, one thing we typically do is also to send Excel sheet where we ask our collaborators to write down the details about the samples, particularly how the tubes are going to be labeled and exact labeling on each tube. Then we have a system in the lab that basically it's kind of a printer that once we transfer the samples to our tubes, we make sure that we keep track of the same label from the Excel sheet and double check that it's the same label in the Excel sheet and in the tube. And then we print it with this printer, and our tubes are labeled in a easier way or to keep track. It's not very sophisticated. I have probably to admit that it's not super sophisticated system, but that's basically our main system. Just make sure that the collaborator writes down the details of the samples, how they would be sent it. Then we transfer these samples to another tubes where we basically put our code in these tubes with a printer. And from that on, I don't know if this is beyond the question, but we start with the project.
Thanks, Oscar. There was something interesting that you mentioned, and it was something that Annie was talking about before we started the webinar, and that's communicating with your collaborators or your clients. Annie, would you mind sharing that example that you had earlier?
Yeah, let me start by saying too that one of the things that I have noted in my career is that the number one reason why a metabolomic study fails is nothing to do with the acquisition on the instrument, right? It is so often all of these things that we're talking about here. How are the samples collected? How are they stored? How is it documented? How is it tracked? You know, did somebody realize that those samples were sitting on a back dock in Houston, in the southern states, very hot in the middle of summer, for 24 hours? Like, these are all important things that are absolutely going to get in the way of a successful metabolomic study, far more than how well did the instrument perform. So I think this topic today is so appropriate because you got to understand the myriad of places where things can fall apart. And speaking to the myriad of places something can fall apart was the example I shared before. As part of great communication that you try to work with your PIs as soon as you can, a PI had come to us and said, oh, we're really interested in running this sample type. We said, hey, we've never touched that sample type before. Can you send us a few experimental samples so that we can just make sure there's no issues, no concerns? Let's make sure everything is going to be okay for you to send those samples to us. Yes, samples looked great. We gave them the full thumbs up, please send us more samples and we'll run your study in that matrix. And then unbeknownst to us, they shift the buffer that the samples were coming in, and the buffer was completely catastrophic to the instrument analysis. And it was ultimately a completely unsuccessful study, and we were frustrated and the PI was frustrated. And it was just this completely inadvertent shift in the buffer the sample came in as compared to what we tested. And again, so important to have such a great line of communication with your PIs, because as I think Oscar said, or maybe Maria, I can't remember now, sorry. There are lots of different levels of investigators, and they sometimes have no idea that a buffer can absolutely be catastrophic to metabolomics analysis. So trying to keep those lines of communication open, even simple things like, this is a great tube type we've worked with before that I think would work great for you, or here's a tube type I would stay away from because it's got lots of environmental contaminants in it, right? Those things can be hugely important to the PIs, and as soon as you can start that conversation, the better. We don't always get that, but if we can, we love it, right?
May I add something in line with these comments? Actually, in line with both what Oscar and Annie said, in most of these meetings with our collaborators, we typically enhance or underline the type of solvents that they have to use. Actually, we provide the solvents if they have, for example, to culture cell lines and then to culture or extract conditioned media and do in their lab, the metabolite extraction. I send my students to work with them in parallel and show them also how to manipulate solvents and pipettes. And it seems that it's a lot, but actually these type of details and the type of like the plastics, like plastic material that we recommend to use, they are like to write down the batch of the reagents that they are using. There's a list of like details that for biologists sometimes they are like minor details, but for us they are so important because they can make a difference and they can provide outliers everywhere based on something like a solvent or changing the plastic tube brand in the middle of the sequence, something like that. And also, yes, I support all this.
I mean, along Maria's point, I feel like there's a lot of education for the people that have to ultimately run the samples that we provide in a way to the PIs, because they don't know metabolomics. They don't know all the things that are involved. So I do think that as much educating of those PIs that we can do to help them understand all of these little things is so important, and just makes for a stronger result on the back end. But it takes a lot of time. And Maria says she actually sends staff to go work with them. I mean, not everybody's got that. These are ideal scenarios, but you do the best you can with what you've got, right? And I think that's the other important part is, while we're all going to be talking about these things that we do, we all understand that everybody's got different capabilities. So it's like, how can you pick and choose the like, oh, wait, that I can do, that I can start doing. I can get that list of plastics. I can get that list of solvents, and make sure that's, because those are really tractable and easy things to put into place in your lab or wherever you're working.
Thank you. So yeah, go ahead, Oscar.
No, I was going to say that we never ask our collaborators or customers to prepare the samples, to extract metabolites, because we've done this a couple of times and it was a disaster. So it's been a long time that we always ask for whatever, urine, plasma, cell cultures, any biological sample, send it to us and we'll do the extraction and the preparation of the sample, because the experience has been very, very bad in this sense. And
the
one
only
more thing
exception
unless you can have a collaboration with students that are working together. So that's, but that's not always easy.
Yeah, difficult.
So that maybe someone
us.
working with you would do the metabolite extraction in your collaborator's lab and combine.
And I was going to say, I would say, Oscar, we do the exact same thing where possible. The one caveat to that is BSL-2 materials. So like we've worked with the DOA, right, and the Department of Defense, and it's like, no, you cannot ship Ebola to us. Sorry. So in those cases, if you're working with highly pathogenic materials, you do need to work with your collaborators to try to find, like, how can you demonstrate that this is no longer a pathogenic sample? Again, all that pre-communication, really so important. Now, that's obviously a pretty rare instance when people are working with super pathogens, but you know, you'd be surprised what things qualify as BSL-2, and just to be aware that, you know, sometimes there are places where you don't want your own staff handling something. So again, that's our only, but again, to Oscar's point, we send us the raw sample because we want to extract it. Completely agree.
Yeah. And then the other thing is the importance of sample collection in terms of what, so we've been discussing, it's very important to meet with your collaborators as soon as possible. But it's critical to know if you're going to meet with someone who has already collected the samples or who is intending to collect samples. And the second scenario, I think, is the best. I mean, meet with someone who still do not have the samples and who wants to collect samples for a metabolomics study. Because my experience is that when the samples have been collected already, when they are in the biobank, in a biobank of a hospital, for example, in general, these samples have been already used for other analysis, genomics, sequencing, mRNA-seq, whatever. And after all these studies, they want to do metabolomics and find out their magic biomarkers when the samples have been defrosted several times, and that's, to me, the most problematic scenario, that samples were not collected to do metabolomics, and they want to do metabolomics, and it's a really challenge. For me, it's been a challenge many times to say, no, we will not do metabolomics with these samples, because it will be a waste of money for you and time for us. And it's difficult to say no to someone who really asks for a service and who will pay for that.
Yeah. So I might ask and dig a little bit deeper. So we've talked a little bit about communicating with collaborators and, you know, making sure that the samples are suitable. So now what happens when the samples arrive? What do you do from when the samples arrive to kind of the day of acquisition? How do you how do you, first of all, how do you keep track of all the samples? Do any of you have a specific system that you use for keeping track of those? I know Oscar mentioned printing the labels and so on, but do you have like a certain file structure, or Annie, I suspect you guys would actually have a LIM system. Maria, I might actually direct this to you first.
We would love to have something like more like a database or like a more, so far we are using Excel spreadsheets with all the metadata, and we have also designs of how the samples are placed in the freezer. For example, each box of samples, we know which sample is in each position in the box with some designs that we have and where it is located in the freezer, because that can also become a nightmare when you start collecting more and more, or receiving more samples, or doing more and more projects. At the beginning, when you start, you have like 100 samples or 200 samples, and it's manageable. But then when you start working on different projects in parallel, you need to be able to rapidly identify where your samples are located in the freezer, and so that you also do not keep the door of the freezer open for a long time, so that you don't affect the other samples or any other things that are there stored. And so far we are working with basic Excel spreadsheets for that, but we want to move towards something more sophisticated, so that this would also facilitate transition when, for example, in our case, we are a research lab in a research center, a small scale, and when a PhD student graduates and leaves, and so to facilitate the communication and how the information goes from one person to another, even if everything is already in these Excel spreadsheets, it would be easier if we can maybe just with the code track all the story of the sample with a huge database. And that's what we are working. We have been discussing this in group meetings, how to improve this for the future. And we also expecting to like expand our lab and our projects too. So
Maria,
far it's something.
Yeah, sorry to cut you off there. I think there were kind of 2 points that you raised that I thought were really interesting. One is that you mentioned scalability, that you have Excel spreadsheets right now, but you're trying to create something that not only works for 20 samples or 50 samples, but maybe 200 samples. And the second key point that I found really interesting was that that traceability across time and between people, and creating a structure that is easily interpretable for other members of the group. So is this something that you would recommend to people, that within their group that they create a cohesive structure for the group that everybody can follow?
Yes, definitely. Yes. Now we all have this same language, let's say, through the Excel spreadsheets, but we want to make it even more like standardized or easier, like, because even if this works so far, we are a small group, so maybe that's also the reason why it's working well. But in like thinking about having more group members, or yes, I would go for something more so to avoid mistakes, because that's the goal, right? We don't want a human error by introducing data into the Excel spreadsheet. Excel always can have, and that's, I mean, that's one of the goals that we have accomplished regarding QC procedures, because we develop a pipeline in Python, and we have like part of the QC monitoring is already in Python in some pipelines that are working very well with everyone. But for this case, we want to think about something else, so that we can not only have like track of like where each sample is located with a code number, and also associate how many aliquots, how many freeze cycles are associated to each aliquot, and who we typically, for example, track who prepares each solvent batch, and when the dates and the person, and identify, and then also track to each aliquot and each sample all the metadata, because that's also a new word. Like even if our lab has analyzed so far around, like the largest study were 500 samples more or less, but for analyzing those 500 samples before we had to curate metadata from more than 1,000 samples. So we had the communication with the medical center that provided all this metadata, and we were the ones that said, no, this sample cannot be included because it's not respecting the inclusion criteria, even if we communicate with, and that links with the previous question, even if at the beginning we have several meetings and we decide some criteria of which samples should be put apart in the freezer, in the -80 freezer in the medical center before they are shipped to our institution. Sometimes they also put apart other samples because they think they may be useful. So then we have to curate all the data before like during the design of the experiment.
Great, thank
Sorry,
you.
I went to different avenues.
No, that's great. Oscar or Annie, did you want to respond or mention anything beyond that?
I was just going to say, so you're absolutely right. Metabolon, we operate a lab information system, right? So we have a LIMS system. So, I mean, gosh, it's almost obnoxious the amount of information we track between, essentially, we log what's called a shipment, right? So a shipment comes in, the very first thing that happens is that sample is received. It automates a response to the client that says, hey, we received it safe and sound, nothing's thawed, all the dry ice is there, thumbs up. Or if it comes in, again, all the dry ice is gone, we want to have a discussion right away to say, hey, what do we want to do now? So again, keeping those lines of communication up with the PI, we then, just like Maria said, we then like pull up, all right, what samples was I supposed to receive? And what we find here, and I think Oscar mentioned this, like the idea of an electronic manifest. So we actually have a templated manifest that we send to say, look, give us your name. How much microliters do you think you're sending us? Metadata that you have associated, you know, any information that they want to include, but then that way we can, when we get those samples in, when we accession them, say, I was expecting 50, these are the 50, this is the 50 they said they were expecting to send us, these were the 50 we received. And then we also log, okay, it's been accessioned, we checked it all. All of that's done on dry ice. We don't want any thawing during the accessioning process. And then we log what freezer it's going into. And essentially, once everything is sort of accounted for, yep, the samples came in, they were healthily shipped, you know, no issues with shipping that we noted, and put into this freezer. And then they go into the queue for acquisition as we go. But that's sort of the checklist of like between when the client is prepared to ship it and when we receive it, those are the sort of steps. And I think Susanna was talking about, like, do you guys have a checklist? And absolutely. Like, there are SOPs after SOPs after SOPs to say, not only is the shipment documented, who received that box on the back dock is logged, when they received it, who was the shipping carrier, what was the tracking number at the time. All of that is a bunch of checklists and a bunch of SOPs that just mandate that we have that. And it's good for things like chain of custody as we start moving into the clinic. Showing that chain of custody of who put their hands on that sample next and where did it go becomes vitally important. And sorry, Oscar, it sounded like you were about to say something too. Sorry.
We share things with your protocols. We don't have a LIMS. We are not so professional. I mean, it makes sense that you have to do it because it's a big company. What I can say is that since we share space with students, with people who are doing research, and people and technicians who are doing services, we have two freezers, one for students, postdocs, and another one only for services. And we also have a system in Google Drive or in OneDrive where we have an Excel sheet, and every time samples arrive and the technician receives the samples, she will go to this Excel sheet, and it's kind of a map of the freezer where she will put the box with the samples. She will write it down in the Excel sheet. It's an Excel sheet that is in the cloud, so it's shared among few people, because for service it's only few people who can have access to this information. Then samples are maintained at -80 degrees until we decide that it's time to prepare the samples for analysis. And as I said, we keep track of where the samples are located now in our freezer. We already have a document by the collaborator indicating the labels on the type of samples, as Annie said, volume, how the tubes were labeled, and yeah, I mean, then we started with the sample preparation, but until that, this is pretty much what we do. We have in place.
I have a slightly funny story, very short, I swear. One of the things, the idea of the electronic manifest becomes so important because, as Oscar was just saying, the idea of a tube comes in and it's labeled. We actually have a term called sample glyphics. Like sometimes you are looking at a tube and you're like, is that an 8 or a G, or is that a 5 or a 3? So trying to get away as much as possible from these handwritten. So that's why so much of that encouraging your investigators to do it electronically, so that your poor lab technicians are not like turning the tube or, trying to figure out what the PI was trying to communicate to us when you wrote it on the tube. Again, not always possible. Inevitably, you're deciphering sample glyphics, but it's something that if you can try to manage through it, it will be so helpful.
So Annie, just speaking on that, we do have our, we have our first question here, and it's directed specifically to Annie. Do you have experience with these 2D barcode systems that are auto scan? Are there?
Yes, they are phenomenal.
Are they? Nice.
So we actually, I mean, I wasn't going to bring this up, but to encourage our clients and our investigators to use appropriate tubes, we will send you tubes. We will send you a barcode reader so that you scan the tube, you put it in, and then when we get back, then all we have to do is scan that same tube. So again, this is about engaging the client to be a part of us in this analysis. So we will send you those tubes. We will send you the 2D barcode reader in a kit. But again, it's also that, and again, now once you get the samples in, now you've got a plate with all 2D barcodes on the bottom. Now you just put that on a reader and then it goes. So now you know the location of those. I know that was very scientific, right? You know where all of the vials are in that rack, and you get that extra confirmation that not only is the tube received, it is in the exact location you specified based on the 2D barcode. So that is some stuff. I wish we got more investigators to adopt that for us, because it is so amazing to give you that feeling of, yes, I know what's happening. Not everybody is as ready to commit to that next generation of sort of technology and barcode reading, but it is a very nice system. And if you can encourage your investigators to use it, it is so helpful.
So we have another question actually on to extend on that, acknowledging that the LIMS systems are expensive to maintain and they probably need IT support, and it's really not affordable for many academic labs. The question here is there another system that's suitable for small groups with limited budgets who have barcoding scanning capabilities, or, is there a good way to use the Excel sheet system? I know, Maria, you mentioned that you use Excel sheets. Our group uses Excel sheets, but is there a way that you can use those and have fewer errors, or make sure that everything is really traceable throughout the study?
So I know there's connections you can make between Excel and a barcode reader. So we do that. So when we accession columns, we've actually gotten it so that we list the serial numbers, and then you just scan it and it populates automatically in Excel through the scanner. So there are absolutely ways to tie your Excel documents, which is a fantastic, if you can't afford a LIMS system, and they're expensive, they're ridiculous. They take an entire team of IT professionals to keep, right? So I agree that it's not an easy solution for your academic sort of work, your academic cores. But you can tie scanners to those Excel sheets to give you that excellent. Now, I am not a technology person per se, so I don't know the exact process, but we do that already in our lab. So I know there's processes that you can use. Maybe Maria and Oscar have actually got more experience with that, but I know you can make that tie between a scanner and it populates right into your Excel file.
We do not have a scanner. We, as I said, our system is not so sophisticated. We basically annotate and keep track of where the samples are in our freezer and who received the samples and the labels of the samples with a cloud system based on Excel sheets. Yeah.
So I'd like to go one step further now. I know everybody has mentioned this is what happens when our samples come in, and this is, you know, how we keep track of everything. And then everybody mentioned day of acquisition or when we prep the samples. So I think this is a burning question that I've had conversations with my colleagues. Is it better to, because often you'll be splitting this into multiple acquisition modes. So is it better to crash that single sample and split it off and dry it down or store it frozen? Or is it better to sacrifice a freeze thaw, split it into aliquots, and then prep the sample on the day of analysis? Maria, I might start with you. What does your group do?
Actually, well, we have many different cases. When we collaborate with medical centers and we design the study from the beginning, so they start collecting the samples, we request that they aliquot when they collect the serum samples from the patients or the healthy individuals. So at that moment, they are already doing the aliquots, preparing the aliquots and storing them at -80. So we receive several aliquots from the same sample, which is great. And then we can just process each aliquot at the moment of the sample prep. The same scenario happens when we are designing a study with collaborators that work with in vitro models. So in that case, typically also we discuss a lot before starting all the process to decide when we want to prepare pooled QC samples also in those cases with in vitro models, because they are challenging in some, if you want to analyze the exometabolome or the endometabolome. And so in those cases, sometimes like you would have to pool after doing metabolite extraction, because otherwise it's challenging and much more difficult. But typically, if we can aliquot before doing sample prep, we prefer. And then one other thing that we have learned with the years is that we try to include in the studies regarding these pooled QC samples, not only technical replicates of a pooled QC sample that can be prepared from a subset of the study samples, depending on how large is the study, or all the study samples, ideally. But also we include process replicates of the pooled QC samples, so that we can monitor not only how, like, the variance during acquisition, and we can use these technical pooled QC samples to correct if there is signal drift or sensitivity drop or something that may happen along different analytical batches, and do some inter-batch correction. But we can also monitor how well was what is the variance associated with the sample prep. And so yeah, that would be my opinion on this aspect. So it depends on the sample, and it depends on like if you are working in a prospective study or a retrospective study, and you are gathering samples from a biobank, as Oscar was mentioning. And of course, we ideally do not want to work with samples that were already underwent freeze thaw cycles. We try to minimize that and do not work with any sample that underwent more than 3 cycles. But that's our experience.
Oscar, what does your lab do in terms of sample prep? When do you prep it, and how do you guys go about that?
Well, first of all, you have to define if you want to do metabolomics or lipidomics. That's the main decision. If it's lipidomics, it's a complete different sample preparation, sample extraction. And if it's metabolomics, we do another one, and then we basically typically combine HELIC chromatography coupled to mass spec, or C18 chromatography coupled to mass spec. And if it's lipidomics, it's always C18 mass spec. And we always implement or execute the same sample preparation protocol for either approach.
And do you do that on the day of analysis or the day of acquisition, sorry, or would you like split off your samples or prep them in any way ahead of time?
As close as possible. Yeah. I mean,
Yeah.
it's not always is possible, because sometimes, as I said, if you have 500 samples to prepare, that will take you more than one day, definitely. And so we extract the metabolites, we put it in the LC vial, and then keep it in the freezer. Not at -80, that's probably not necessary. You can keep it at even at 4 degrees or -20 until all the samples have been prepared, and then you start the collection of the data. But if the cohort of the data set, the set of samples is small, tens, let's say tens of samples, yeah, we typically prepare all of them, and then we start the acquisition, the data acquisition right away.
Great. And Annie, what does Metabolon do, if you don't mind us asking that detail?
Yeah, no, of course. So for the bulk of our work, we extract the day before it's going to go on the instrument, and then we leave it dry overnight to just in like a nitrogen dry box. And what I was going to say is I was listening to Marie and listening to Oscar, and unfortunately, this is one of those terrible answers where it kind of depends on what you're working with, right? There are some samples that are way more sensitive to freeze thaw cycles than other. So ideally, what you're going to do is do some validation in whatever matrix you're working in heavily and sort of say, how many freeze cycles do I have to play with in my process to know what is safe and what is danger zone? Consistency is always so important. If you have to deal with samples that have been frozen and thawed 5 times, as long as everything has been frozen or thawed 5 times, data shows us that even though it's not going to necessarily represent exactly what that sample looked like after one freeze thaw, you can still draw really accurate biological conclusions because things tend to degrade in a similar fashion. But at the end of the day, every matrix has got to be handled maybe a little bit differently, and even different sort of end cases. So there is one, our clinical samples, for example, we have to process and run them on the same day. So if we have the doctors that are sending in those samples, our SOP says process, run same day. And that's just because that's clinical. That's like, there is no room for wiggle. We got to get a turnaround time back to those doctors in 8 days. So for that particular product, we do it one way. But for our standard method, we have validated that an overnight dry nitrogen dry box, the samples are very stable. And we actually have a time course. Like, all right, what if I had to keep them in that dry box 2 days? What if I had to keep them in that dry box 3 days? Again, at what point do I start degrading my sample? So if there's any question, just do some tests. Those are things you can do in your own lab. Buy a pool of plasma, buy a pool of urine, just set up some tests. And then there's no question. You kind of know what the best for your method, for your sample type is. And I get that that takes some work, but that's how you're going to feel super comfortable that the data you're generating is going to be the best quality.
Oscar, go ahead.
But I have to say, so I may agree that you can conduct a study with samples that have been defrosted 4 times. But the problem in my experience is that it's difficult to find that all the samples in that study have been freeze thawed 4 times. Because typically clinical samples, they come from different hospitals, different places, and you may find that the sample from this hospital has been defrosted 2 times, and from this other hospital 1 time, and from this other 3. And at the end, the cohort is based on diseased people, healthy people from different places, and that when there is a big problem there in my experience. So.
And maybe I wasn't clear. I meant like if everything was consistently frozen, thawed 5 times, right? Like, you're absolutely consistency, gosh, in everything we do in metabolomics, same tube type. I think Maria mentioned earlier, like, don't switch tube type lots halfway through. It may seem sort of like this no big thing, but oh no, that can have a big impact. So consistency. So sorry, and if I wasn't clear earlier, yeah, I mean, my point is, don't compare a 2 freeze thaw cycle to a 4 freeze. You're going to see differences in the data that is solely a result of the freeze thaw cycle and has nothing to do with the biological intent. Consistency is key. And sorry if I wasn't clear about my statement earlier on that.
So here's another question that comes across a lot that I've had discussions about. So you would have designed your study, your sequence, you're prepping your samples, everything's ready to go. You have your tubes labeled and everything. And then let's say there's a sample where you don't have enough volume, or it's hemolyzed, or there's something wrong with the sample. Let's say it's all coagulated. What do you do then? Do you take that out and readjust your sequence on the fly, or do you pop in a QC and exclude the data after? Does anybody have any recommendations for what to do at that stage?
Ask your PI. Communicate with your PI and then ask your PI. So in all honesty, in most cases, hemolyzed samples, we will document the hemolization, but we will still run it. Coagulated samples, that's a very different story because, hemolyzed sample, you're going to see signatures of the red blood cell contents in a way that you don't. It's not going to damage your instrument in any way. So you can still run that sample and simply flag the data on the back end and make sure that everybody knows the sample was highly hemolyzed. And again, we have a limb system, so it's associated with but regardless, that's an important piece of information because that will absolutely change your statistic. But there are some samples. I remember one example where we had a sample come in that was so, the client must have had some sort of severe case of lipidemia. They couldn't pipette the plasma. It was so thick. They said it was like jelly. It was like, okay, we need to reach out to the PI and look, we can physically not handle your sample. What do you want us to do with this? We could potentially try to dilute it. We could homogenize it. Either way, this is not going to be treated like the rest of the population. In extreme cases like that, we would generally remove it because it's not going to be representative, but it's important to still let the PI know that this particular sample was a really unhealthy individual, and that may be important for you to know in your study. Or we can try to figure out a way to get it into the study, dilute it. But again, it's going to have all kinds of issues on the back end. But again, a lot of that is for us dictated by, what does the PI want to do with that? How do they want to handle that?
Oscar or Maria, do you have any comments on that?
It's pretty much the same. I receive the feedback from my technicians and we decide together what to do with this type of samples. I have to say that it's not very common. Sometimes, the most common situation is that the tube contains less volume than expected, and you plan to extract, I don't know, 50 microliters of whatever plasma, and there is less than 50. We always ask for more. If we use 50, we always ask for more than 50. But sometimes it might be less than 50. And then so we readapt the extraction volume to less starting material. But other than that, if it's more problematic than that, yeah, we have to contact the collaborator and then say, look, what do you want to do with the sample?
Yeah, I agree with Oscar. The same happened to us with you expect a certain volume from a collaborator that indicated you will have 60 microliters, and then when you thought you cannot even pick 50, and so in that case we scale it. So we change the volumes and we scale it and we include it in the study, mainly if they are precious samples from animal models that or something that it's important to have the data and see, then evaluate or of course write that down. And that's where we always come back to write every detail down, because this may be important when you do data analysis. And another example that I recall, when in a study in which I worked with samples from patients having cystic fibrosis, and this was exhaled breath condensate. And so when we were after freeze drying the samples, we found out that the sample had a huge amount of salt content, and that was excluded just to avoid damaging or even clogging the electrospray ionization source. It was impossible to like even applying a huge dilution factor. We would like not run it just in case, but that's another example that came to my mind now, that is not related to serum or like a typical matrix.
Thanks, Maria. I might also just jump in from a perspective that we do in our group. And this is something that came from a large scale study that I did several years ago in Craig Wheelock's lab. And I was working with Hector Gayart Ayala. And I remember we came up with these SOPs and everything was well documented. We had a very good plan. And then one day Hector turned to me and he said, "Stacy, today we're not humans, we are robots." And the idea was that from now on, we're not going to change anything. And so if we had a problem with a sample, we weren't going to adjust anything on the fly. We took out that sample, we popped in a QC, we recorded it. So then that way we weren't adjusting anything. There was no chance of anything getting mixed up with the sequence, with the numbers, with the spreadsheets that we had. And all we did was in the file name, we just wrote exclude. And so we knew then to keep track of that, and that was going to be a QC, and we would exclude it later. So that might be something else that other people might find helpful as well. So we're approaching the hour here, so we just have a few minutes left. So I thought that I would pose a question. I'm not sure if anybody has any horror stories of what can go wrong. So if you don't have a good system in place, if you don't have SOPs, if you haven't been careful, what can go wrong? Does anybody have anything that they want to share on that, or any horror stories?
Yeah, I can share one of my first experience during my postdoc. I don't remember how many samples we analyzed, and at that time we didn't care too much how the samples were collected. We found biomarkers, and everybody was super excited about what we found. And at the end, it turns out that it was an artifact of just the fact that some samples were collected in using one protocol, the other ones were collected using different protocol, and the differences were just artifacts. And it was a waste of time, money, and excitement in both sides.
Maria or Annie, do you have any words of wisdom or horror stories to share on that note?
Oh, I thought Maria went unmuted, so I thought she was going to go. Sorry, I was going to say, gosh, the horror stories abound, and I think that's the problem, right? And I think all of us have, like, God, as you said it, I was like, oh, well, I could tell them the mouse chow study that was, again, it's so similar to Oscular, where like the control mice were fed blue chow versus the treated mice were not blue chow. And so the PI was so excited because when we processed them out, there was like, oh, these massive, amazing biomarkers. That was blue dye number 95, right? So much of what is going to just floor a successful metabolomic study is sample design, tubes that you put it in, all these different things that, again, may seem small in the process of putting the study together or getting it shipped or how many times it is freeze thawed, are just catastrophic or can be catastrophic. And the examples, you can think of so many possibilities. So much of the lesson here is just consistency, try to bring as much consistency, try to keep dialogue open. Again, and I think, again, forgive me, I'm terrible. Maria or Oscar talked about establishing a list of good solvents or good salts, or, oh, whatever you do, please don't put Triton X anywhere near your system, right? Like all these little like things that you can just send to people so that they know it, so you can try to bypass some of these things. But horror stories abound from the myriad of ways people can mess up their samples before you try to acquire them.
Maria, did you have anything to add?
We had a case in which we analyzed the endometabolome of cell cultures in vitro models, and actually we identified a confounding factor introduced by the culture media just because we were generating a pretty large number of samples around, like for in vitro models working with around 70 samples. It's quite a lot because you have to plate all the samples in parallel and try. And based on the design of the experiment in which we use each culture media batch for culturing different cell lines in parallel, we were able to identify that there was actually one batch that was anomalous, and then we were able to track back and understand that actually it was prepared by someone else in the lab, in my collaborator's lab. And so, but based on the design of the experiment, that each culture media batch would be used to prepare different cell lines in parallel, we were able to discard those samples that were cultured with that particular cell culture media batch, and we were able to continue and analyze the rest of the data. So that maybe highlights why it's important to write down everything and to transfer this information to the collaborators, because in many cases they never experience this type of implications, right? They think that changing from one solvent to another or one brand to another may be the same, and it's not the same for us.
Yeah,
I was
great.
going to
Thank
say,
you.
like, I'm suddenly very aware that we might be scaring anybody away from ever trying metabolomics with all the discussions that we've had. So I did also maybe just want to say as an end note from my perspective, I'm actually also really surprised at how robust sometimes biological signatures can be. Even if there's some confounding issue, often that means you're going to lose the smaller biological significant signatures, but you're still going to be able to capture the bigger ones. So I don't want to scare people away to say, yes, there's so many confounding factors, don't even try. Like, no, what these confounding factors will often do is kind of make some of the smaller changes disappear. They won't become statistically significant, but you'll still be able to see those really big changes. So it's not so scary, but there are things that you can do to be able to see even those really small biological factors by controlling these things. So I just wanted to, it's not that bad. It's really not that bad.
Thank you, Annie. So I think we've run out of time for today. I do see that there were a few questions in the chat that we weren't able to get to. There were a couple of questions about freeze-thaw cycles and sampling and confounders and stuff like that. We have had a couple of podcast sessions on study design and sample collection. So for those of you who are interested in that, we do have them posted on the Metabolomics Society's website. And there were a couple of questions about data acquisition, time and autosampler and stuff. Later this year, we will host a podcast session on QA/QC and also data acquisition. So I'd encourage everybody to keep an eye out for those. So at this stage, I would like to thank everybody for attending today. I would like to thank our three panelists. Thank you so much for your time, for your expertise, for the great discussions, and a big thank you to Susanna for helping field the questions today for us. Thank you, everyone. Have a lovely day.
Bye-bye.
Thank you very much.
Bye.
Bye.